Professor Steve Austad, thank you so much for joining us today.
My pleasure. Happy to join you.
I'm keen to start with a lightning round, mostly to warm up my brain while the caffeine kicks in. And I'm going to ask you to try and do what no scientist has ever been able to do and keep your answers to a single sentence or less, if possible. The first question I have for you is, what is the geroscience hypothesis?
The geoscience hypothesis is that by targeting the biology of aging, we can delay or eliminate multiple diseases and conditions of later life at the same time.
You've written many books, but I believe that your most recent book is Methuselah Zoo. Who supposedly was Methuselah, and what do you mean by Methuselah Zoo? You might need 2 sentences for that one.
Methuselah was reputedly the oldest person recorded in the book of Genesis, 969 years old, and he's typically used as an icon of extreme longevity. So since my book was about extreme longevity in the animal world, I thought Methuselah Zoo had a nice ring to it.
You and Kathleen Fisher came up with the Longevity Quotient. What is the Longevity Quotient?
Longevity quotient is just a simple way to assess how long animals live relative to other animals of the same or similar body size. And that's important because body size is a key to longevity in one after another animal group. Larger species typically live longer.
And what is an average longevity quotient, or LQ?
An average would be 1. 1.0 would be your absolutely the exact longevity that an average mammal, that's confined to mammals, of your size would be.
And which familiar animals have a longevity quotient of around 1?
Dogs is about 1, has a longevity quotient of about 1.
Who was Ming the quahog?
So Ming the quahog was a clam that lived 507 years. And in fact, could have lived potentially much longer, but he or she was actually killed by a scientist who was studying something unrelated and only later discovered that Ming was so old.
You said that the LQ is for mammals only, but do you have a rough idea about what Ming's LQ would have been?
The reference values are all for mammals, but if you assumed that it would probably be around 50 or 60.
And which vertebrate has the highest LQ?
The birds that have the highest LQ are ocean birds, shearwaters, and they have LQs of about 6. But there's an important qualification about that. LQ has reference values that are all from animals in captivity, either in zoos or in research, whereas the bird data are all from birds in the wild. And the wild being a much more hazardous place to live than a laboratory, you can imagine that these LQs would be substantially higher if the birds had been protected in a laboratory.
Which vertebrate has the highest LQ?
That's a good question. Probably a salamander, a blind cave salamander, because they're very small, but they live probably a century or more. By very small, I mean 12 inches, 20, 25 centimeters, 30 centimeters, and it takes them decades and decades and decades to reach even that size. Everything is slow about them. They grow slowly, they reproduce slowly, but they live a long, long time.
And how about fish, Steve?
Yeah, fish, the longest life fish that we know about is the Greenland shark, which, if the way that we assess their age is reasonably accurate, can live up to almost 400 years. But we're not quite so sure of that because the way of assessing their age is rather indirectly. With the clams, they have their birth date in their shell, and you can tell from the growth lines in their shell when they were born by counting the growth lines. You can't do that with Greenland sharks. So we had to use a different way to estimate it. And that estimate has a lot of potential error in it. It could be off by as much as 150 years. But even if that's true, that would still make them living up to 250 years, which would probably still make them the longest live vertebrate that we know about.
Which mammal has the highest LQ?
Mammal that has the highest LQ is a bat. It's called Brandt's bat. It weighs about 6 grams, and it lives up to 41 years in the wild. So if you consider for something as tiny as a bat, how dangerous life in the wild could be, that's really remarkable. That translates into an LQ of about 10. So it lives about 10 times as long as an average mammal that size would live.
And if we assume that maximum human lifespan is around 100 years, what is the human's LQ?
Humans are about 4.5. The reason that I use 100 years, even though we know there's someone that's lived 122 years, is that we have billions and billions of birth records from humans, and we're comparing them to animals that we might only have a few hundred or a few thousand birth records. And the more records you have, the more extreme values you're likely to get.
I was going to say the chance that Ming is the oldest quahog that has ever lived seems vanishingly small.
You're right. You're absolutely right about that. But we've aged enough quahogs to note that Ming is very long-lived, upper at least 1% of all quahogs. But again, if we measured 100,000 of them, I'd say we're very likely to find some that are even older. And especially since we don't know how long Ming would have lived. Remember, Ming was killed by a scientist. He didn't die a natural death.
You made an interesting wager with Stuart Olshansky. Has the first person to live to 150 already been born?
That bet came about because a reporter asked a room of geroscientists like myself, when will the first 150-year-old person be alive? And I said, I think the person's already alive, probably very young, but already alive. He read about that in the press. I had a book that had come out not long before. He had a book that was coming out. He thought this would be a nice bit of publicity for our books, and it has proved to be, as people are still talking about it. We made the bet in 2001, and we're still getting asked about it quite a bit.
Could be you, Steve. That would be so cool.
Yeah, and in fact, we're both, even though the oldest person to ever live died in 1997, and no one has approached her age since then, I'm still convinced that I'm going to win. And he, of course, is also convinced that he's going to win. So we doubled our bet in about 2016.
Do you know Franz de Waal, the primatologist?
I do, in fact. The late Franz de Waal. Yeah, I know him quite well.
Oh, cool. He wrote a book that I love entitled, Are We Smart Enough to Know How Smart Animals Are? Do you think we are?
No, I think we're constantly being surprised by things that we didn't expect animals to do. Now, having said that, I think scientists are a lot more hesitant to believe things that animals can do than the lay public. So, I used to train lions for a living. I spent years and years and years kicking around the bush with various kinds of wild animals. And when someone says, you know what, wild birds could actually recognize the song of their neighbors and distinguish it from the songs of other birds, that doesn't surprise me in the slightest. Of course, it would be very valuable to be able to do that. So, of course, animals can do it. It's something with a brain as small as a honeybee, can understand symbolic logic because it's useful. So, one of the things that Franz de Waal shows is this video of a chimpanzee doing a memory test that's absolutely trounces the best human memory experts and does it again and again and again and again. So for some specific reason, it became important for chimpanzees to have this incredible spatial memory, and they have it. It is much better than the humans' spatial memory.
What pets do you have, Steve?
Let's see. Right now, we have 4 dogs, 3 cats, and 2 parrots. But this is kind of a low ebb for us right now. We've had as many as 8 dogs, and 8 cats, and a whole variety of exotic animals from pythons to hedgehogs to coyotes and raccoons. Yeah, my kids grew up, they never knew what they were going to run across in the house or in the freezer, if a pet died.
Other than being mauled by a lion, what's the most uncomfortable interaction that you've had with another animal? And by that, I don't mean a human.
Except for the time I was mauled, I had a number of very close calls that I didn't get hurt at, but which it's almost miraculous. So one time, there was a mistake at the place that I work. Now, lions get fanatically possessive over food, even well-trained ones. And I walked into a compound that had a loose lion in it that I didn't know about with a big handful of horse meat. And when I realized that there was a lion loose in there, and it was an area where there were no trained lions, there were only wild lions, I threw the horse meat as far as I could in the hopes that the lion would run after the horse meat, but it didn't. It just continued to stare at me. And so I edged gradually, trying to get close to the door of the compound. And when I got close, I bolted for it. At the same time, the lion bolted for me, and I managed to slam the door in the lion's face. And, I was perfectly fine, but I had to go sit down for a while because once the adrenaline wore off, my legs got a little weak and shaky for that. And I mean, I had others. One time I tripped with a lion was chasing me, and he actually came over and put his mouth on my stomach and didn't bite, and then just turned around and walked away. And I was on the ground, so I was totally helpless. And then, oh yeah, there was a great time when I stepped on a small crocodile uh, in the water. It was in water about up to my knees, and this animal was in the mud, so I didn't know. And I stepped on its back, and as soon as I did that, it took off and jerked my legs out from under me. So I went face down into the water that had a crocodile, and I was just imagining the crocodile is going to be gnawing on my face, my fingers, and everything. And fortunately, it was long gone. But again, it was one of those times. I think I got up out of the water and must have run 100 meters before I even, took a breath. So yeah, I've had more than a few close calls, I'd say, with animals.
Sounds like you've been quite lucky to make it to your 70s, Steve.
Yeah,
Remind,
I feel like I am. Yeah.
what's the most unexpected thing that you've seen an animal do?
I think the most unexpected thing I've seen an animal do is a bird attack another bird of the same species and kill it. This is a small wren. These wrens eat insects, and so you don't expect them. You expect them to maybe have a territorial tussle, but you don't expect one to get the other on the ground and peck it to death. But I saw that happen. It was very hard for me not to break it up, by the way, because these were birds that I knew well. I had them color banded, so I knew each individual involved.
If every biologist who studies actual animals had to spend at least a couple of months in the field studying animals in their natural settings, would research into biology improve?
I think it would become a lot more realistic. For someone like myself with a field biology background, when I look at the way that we keep our animals in captivity, particularly the research animals, we keep our mice in a shoebox size cage their entire lives. We feed them the same thing they have the same light cycle every day. They have very little sensory stimulation, and they're protected from any exposure to microbes. The mice, let's say, the mice that we've given a certain diet or manipulated genetically or given certain drugs, those mice might live longer in the laboratory. In the field, they would they would be dead in short order. So I think that this actually matters because let me give you an example. Some friends of mine did some research that they were using older mice. They took their older mice, these are laboratory mice, they put them in a cage that previously had held healthy pet store mice. and within 2 weeks, they were all dead. The exposure to the normal microbes that pet store mice live with was fatal to them. So that suggests that the ways we know to make animals in the laboratory live longer may or may not have much relevance to making things outside the laboratory, whether they're our pets or ourselves, stay healthy longer.
If you could bring back a single extinct animal, what would it be?
Yeah, that's a great question. If I had to bring back an extinct animal, I think I would bring back one of the large pterosaurs, these flying reptiles that could get as large as giraffes. I would love to see one of those, and I would love to see it fly.
A pterodactyl is an example of those.
Yeah,
Familiar
that would be
example.
a small example, right?
You've worked in many very beautiful and remote places. Are there one or two that hold a special place in your heart?
Yeah, the place that had the most unique animals that I love for that was probably New Guinea, but it was also one of the hardest places to live. The places that I treasure for just being able to watch animals do amazing things is the East African savanna or the Southern African grasslands as well.
My experience has certainly been that the most special places are often the hardest places too. And I don't have anything like the experience that you have. If I think about those places, then Madagascar is one of those places that comes to mind.
Yes, Madagascar. I agree, but I've only spent a couple of weeks there, so I don't feel like I'm as familiar with it. But yeah, every animal there is sort of unusual in its own interesting way.
You mentioned scuba diving a few times in Methuselah Zoo. Is there a particular place that you've been diving that you've especially enjoyed?
Surprisingly, probably the place that I liked the most was the Chuk Lagoon in Micronesia. The reason I like that so much is there are all these World War II ships that were sunk there, and I never thought I would like diving on the ships because I'm a biologist and I like the flora and fauna. But the first time I went down about 40 meters and sort of settled on a reef that I thought was a reef, it turned out it was the deck of the ship that had become a reef because it had been underwater for that long. And then there was something surreal about swimming next to a ship that had a big hole in its side with airplanes still in the hold. And so, going into the ship and going into the surgery suite, that was a lot more fun than I ever anticipated. In terms of the most memorable fauna was probably in Hawaii, where I was in a place where you could probably see 60 or 70 sharks at the same time.
Yeah, I think Micronesia probably has about the highest density of world-class wrecks in the world.
Exactly. And those are surprisingly fun. I never thought I would really appreciate those, but I did.
For the general public, in the context of our modern society and trends in disease, which candidate gerotherapeutic or anti-aging drugs or supplements do you think have the most potential?
From what we know right now, I think Rapamycyin has by far the most potential. That's probably because it was the first drug discovered that made mice live longer, 2009. But since then, we've discovered so many things that it does besides make mice live longer. It's good for cancer, for their heart, for their immune system, for their balance. And we just keep discovering new things. Now, having said that, the new GLP-1 agonists show a lot of promise. And I might almost give them the advantage because the promise that we've seen in those has been in people, whereas everything I just said about rapamycin has been in mice. And translation from mice to human is rare.
Yeah, I completely agree. I think that semaglutide and tirzepatide and some of these other drugs probably have more potential. But would your response differ if we're thinking about people with healthy lifestyles? So people who are already in good health, so the general
Well, that
public,
is
average person out there.
yeah, the people with very good lifestyles. That's very interesting because this is something that we don't model at all in animals. We don't try to combine the best lifestyle with our drugs or our diets or anything. And it may be that if you're living the best lifestyle, none of these things can help you that much.
Yeah.
We don't know if that's true or not, but it's certainly a theoretical possibility. We know, for instance, that a lot of the drugs that benefit older people don't benefit younger people, and that's because they're already operating at such a high level that the drug is not doing anything for them.
I'd like to come back to that because I suspect that some of the better gerotherapeutics, including rapamycin, might actually have some effects that oppose those of a healthy lifestyle. And that's been well demonstrated for metformin and adaptations to endurance training and resistance training. With rapamycin, there are reasons to think that it could, for example, blunt some of the adaptations to resistance training. We know that it reduces mixed muscle protein synthesis in response to overload of various forms. But watch this space. And then, final question of the lightning round. What's one thing that you wish that the general public better understood about aging?
I wish that the public understood better how important their lifestyle habits when they were young are for determining how healthy they will be later on.
Speaking to someone who's just injured his lower back again. So I certainly relate to that. And I'm conscious of the fact that I need to avoid bad musculoskeletal issues because they will come back to haunt me. So thanks for that, Steve. I'm quite keen to hear a little bit about your entry into geoscience. And I know that it could be a long story and the subject of the great American novel that you're still yet to write. But I was wondering if you could give us just the potted history of that entry and maybe up to the point where you began studying opossums.
The entry really is with the opossums, which I was studying, but with nothing that had anything to do with aging. But the study I was doing, I needed to recapture them once a month and examine them. And what I discovered in doing that is within 3 or 4 months, they would go from being very healthy, prime age adults to being old and feeble, with shrunken muscles and cataracts and things like that. And it was such an unexpected observation that I just got fascinated. Well, why don't they live longer? They're about the size of a house cat. Why don't they live as long as a house cat? And that question really drove my research for at least the first 10 or 15 years that I was in the field. The original study that I was doing, by the time it was published, I'd completely lost interest in it, and I was on to aging. And that also made me wonder about our knowledge about how long animals live, because when I did hundreds and hundreds of opossums I tracked from birth to death, and none of them ever lived as long as 3 years. And yet when I went to the literature, when I got back to the US, I found, opossums live 7 years. And I thought, well, why do people think that? And it turns out that was just an error, some bad science, superficial science. But it made me question now whenever somebody tells me, oh, I have the dog that's the oldest dog that ever lived. I really want a lot of evidence before I'll believe that.
Prior to studying opossums, what are some of the things that you'd done in your adult life up to that point? Because you entered this field relatively late, and certainly by contemporary standards, very late. Because I think a lot of people nowadays, they feel that if they don't have academic tenure by the age of 40, then they don't have a chance anymore.
Right. Yes. So I was unusual. So between my undergraduate degree and when I started graduate school was 6 years. 6 years during which I was trying to write the great American novel, but at the same time, to write the great American novel, I was accumulating experiences. And so I had a lot of odd jobs. I drove taxis, I worked as a messenger in New York. I hustled pool for a living for a while. I was a newspaper reporter. The last job I had was training animals for the movie business, and that awakened really an interest in animals that I'd had all along that I didn't really think about, and got me to start taking science classes and to eventually go back to graduate school. But I started graduate school when I was 30 years old when I started graduate school. So by the time I got my PhD, I was 34. So yeah, that's actually a pretty delayed career start for an academic.
And if we start to focus now on the biology of aging, to put it in simple, easy to understand terms, how would you explain the biology of aging to a 12-year-old?
In the simplest terms, the biology of aging, I think, can be explained if you think of life as being driven by a slow, highly regulated fire. So metabolism is a slow, regulated fire. It's exactly the same chemistry that you get when you burn wood. And we know that fire has unintended or not useful side effects. It has smoke, it has cinders. The smoke and the cinders and the unanticipated side effects are what causes us to age. It would certainly be advantageous if we didn't age, but I think it's not compatible with the fundamental biochemistry of life, which is inherently destructive.
I like your use of analogy, and there are many theories of aging, and your answer might also map to the analogy that you've just made, but are there one or two that particularly resonate with you that have been put forward?
Yeah, so when you talk about theories of aging, I think it's important to distinguish theories, which are broad general explanations, from hypotheses that tend to be very specific mechanistic explanations. And I think the important distinction is the theories, only one is going to be right, but the hypotheses, many could be right simultaneously. So someone said, well, aging is due to DNA damage, unrepaired DNA damage. That's quite feasible, and there's a lot of evidence to back that up. Someone else said, well, aging is due to damage to proteins and for the tendency of proteins to clump together when they're damaged. Well, they'd say, okay, well, that probably plays a role as well. So when I first got into the field, I read a paper that categorized over 300 theories of aging. But I looked at them, first of all, most of them were not theories, they were just hypotheses, but a lot of them could be true simultaneously. It made me realize that it's beguiling to imagine aging can be reduced to some simple terms. That doesn't mean that we couldn't intervene in it in a simple way, but the process itself is incredibly complex. And I think that's something that a lot of my colleagues overlook because they don't realize, for instance, a drug, let's imagine we have a drug that targets aging successfully. The idea that you could do that without having side effects, without affecting other things that you didn't really intend to, strikes me as naive. And I think that virtually all these things that we now think are addressing aging are going to have unwanted side effects. And so we may end up doing drug combinations, most of which are to counteract side effects of other drugs.
I'd like to switch now to focus more on comparative biology. And as a comparative biologist, you've been pushing the geroscientific community for some time to study long-lived species. But meanwhile, a lot of geroscience is just focused on a handful of species, in particular fruit flies and yeast and the C57 black 6 mouse, usually housed in specific pathogen-free conditions. If we focus on the latter, then what are some of the shortcomings of using those particular mice? I've heard you say that your colleague has referred to them not as mice, but as mouse-like creatures.
Yes, mouse-like objects. First of all the animals that we study are demonstrably unsuccessful. at combating basically the destructive processes of aging. That's why they're all short-lived. They all fall apart quickly, even for their own group. Mice do this really fast for a mammal. Fruit flies do it really fast for an insect, and the worms we study do it really fast for them. To me, ignoring the fact that nature has had several billion years and billions of species to experiment with for better ways of combating aging, it strikes me that those are the species that we should be focusing on, because humans are already very successful at aging. It takes us decades and decades to age. We're the longest lived of the terrestrial mammals. So what we discover with our own ingenuity to make a mouse or a fly or a worm live longer is, I think, very unlikely to also make something that's so successful like us live longer. Now, that doesn't mean I think that those studies are useless. I think what they've taught us is the places we want to look. They've taught us about DNA damage. So we want to look in these long-lived animals at, well, how do they combat DNA damage? They've taught us about proteins aggregating. Well, let's see how the long-lived animals do it, rather than relying on our own ingenuity. Now, our own ingenuity could come up with something fabulous, but nature is smarter than you are. That's the way I tend to think of this, is that nature has come up, because it's had so much time to experiment, nature has come up with solutions that we're very, very unlikely to think of ourselves.
You've alluded to the fact that the determinants of longevity in wild animals and captive animals aren't necessarily the same. When you were speaking about birds earlier, what are some of the phenotypic differences between animals in the wild and captive animals if we focus on those lab species? Are there some that are relatively consistent across different animals?
The clear pattern in nature is that animals that, by virtue of their intrinsic capacities or the environments they live in, tend to live in a safe environment, evolve very effective anti-aging mechanisms. And it's easy to see why this would not be true in short-lived animals. So mice, for instance, live 3 to 4 months on average, and maybe up to a year in the wild. So nature would not have any reason to favor a heart that lasts 40 years in an animal that's likely to be killed by a cold snap or a predator over the course of a few months. However, if an animal is in an environment where it quite conceivably could survive external accidents for a longer time, then it becomes useful to have a heart or an immune system or a brain that resists aging. And one of the interesting patterns in the birds is that the longest lived birds live in the safest places. They tend to live on islands in the middle of the ocean, and they tend to not even land on those islands except in the dark when they can't be seen. Or they live in caves, or they live in the deep ocean, places that are very stable and places that are very safe. I think there's a huge bestiary of animals out there that we don't even know anything about yet, that might have secrets to teach us about aging that, again, that we're unlikely to discover by studying only these short-lived animals like flies and mice and worms and yeast.
And I'll just emphasize something that you drill home in your book, which is that flight is really helpful because you can relocate to better conditions. And think about that in the
Exactly.
context too, because people in many parts of the world, they don't have the resources to move to more favorable environments. Whereas if you have the finances that permit that, then that can have a dramatic bearing on your longevity. We were speaking before we started recording about the fact
Absolutely.
that the Philippines and life here is a lot harder than it is back in England. There are so many things that can go wrong on a regular basis. I know that you've got some first-hand experience of skirmishes in the wild and things that might have appeared to be quite benign, small cuts and things that nearly have got you unstuck.
Yeah, I once had a conversation about this with Jared Diamond, who's also spent many years in New Guinea, about
Written some
just
great books.
how life on the edge it is in New Guinea. The people that I worked with in the village, very few of them lived into their 60s. It was from a variety of things. It was from accidents, it was from violence, it was from infections, small things like we took aspirin with us. Aspirin was a miracle drug. These people who were living with malaria, chronic malaria, you give them some aspirin and they think that you just gave them a miracle drug. And it's something that we don't think about. And when I got really sick several times from small infections. In fact, I had antibiotics with me. And these bacteria that were infecting me had never seen antibiotics because so the antibiotics were fabulously effective at killing those things. But again, the people that live there can't get out, can't get medical attention. We one time had somebody that a tree had fallen on her and broken her leg, compound fracture. And she wanted me to fix it. I said, no, I'm not that kind of guy. I can't do that. You have to get to the airstrip. You have to get an airplane and you have to get to a hospital. Otherwise, you're going to die. And that was a simple reality.
I wasn't intending to discuss this. I'm also not that familiar with the primary research on it, but I know that you've done a little bit of work looking at Apo E alleles and their prevalence in Papua. And my understanding, and it's a superficial understanding, is that their potential effects on risk of dementia might interact with environmental conditions. And when you're in an environment where there's a high pathogen load, for example, having the ApoE4 allele might actually not be so detrimental. And so if the prevalence of those alleles is high in those places, it might make some sense. And whereas in the Western world, where we've got sanitation and the conditions are generally much more hygienic, the ApoE4 allele might quite strongly dispose people to Alzheimer's disease. In that part of the world, it might not be so problematic. And so it's an interesting example of how someone's genetics interact with the environment, which I think is very relevant to what we're discussing today.
That's an excellent example of exactly how this is also an excellent example of the fact that genes or drugs or other things have multiple effects. You can't say they just do this one thing. ApoE4 predisposes to Alzheimer's disease and to cardiovascular disease in certain environments. And in other environments, it doesn't. It actually is protective against pathogens. So I think this is one of the problems with people that are looking for longevity genes in humans. People that study 100-year-olds saying, what is genetically unique about them? Well, they always find something. But the thing is, if they're studying this group in the US and they're studying it in China, they tend to find different genes that are involved in it, which makes perfect sense to me because, over the last 100 years, the things that people live through in the US and things that people live through in China or in other parts of the world are different. And then today's environment is different in those places. Again, it's one of the things I think we oversimplify is, this is a good gene. This is a good allele. I like the way you said allele because we do tend to conflate genes and alleles, and they are different.
Just on that subject, Steve, has anyone tried to systematically look at geographic variation in candidate longevity genes and said that, well, in this part of the world, in these conditions, these map to greater longevity, whereas elsewhere, these particular variations are more favorable?
No, and that's interesting. And I think it's coming because it used to be that there weren't that many of these centenarian projects, but there are more and more and more in more places. The trouble is the groups that are studying them in different places don't talk to one another specifically. They tend to want to hoard their information for themselves, and that's unfortunate because what we would learn a lot from exactly that type of study. Like I say, it's coming because more and more, and this is really pioneered, I'd say, in the UK by the UK Biobank, where people basically donate their own information. You know, hundreds of thousands of people donate their own information, and now these biobanks are arising all over the world. I just came back from Saudi Arabia, and there's a biobank of 180,000 people there already.
Oh, wow. Yeah, there aren't many things that make me proud to be British, but the UK Biobank, as a nerd, is one of the few.
It certainly is, yeah.
Just on the subject of centenarians, there are many things that have cropped up in geroscience over the years that have subsequently been shown to be dubious. And among those, we have the blue zones. And Saul Newman, of course, recently won the Ig Nobel Prize for his work debunking the blue zones. And he basically has shown that these blue zones, where there's a disproportionately large number of centenarians, are places that are characterized by dodgy birth certificates and pension fraud much of the time too. Do you think there's anything to learn from the blue zones? I suppose that the principle of studying places where people do live a long time and trying to understand something about their lifestyles does make some sense. I'd argue that it might make more sense to look within a country at where people live an unusually long time. So if you look at the US, for instance, there's massive discrepancy between people who are living in Marin County in California, where they live a very long time and they're very affluent, sure enough, and people who might be living in parts of the South where life expectancy is much lower. But that's quite different from this approach of the blue zones that was taken.
Yeah, so let me defend the blue zones to a certain extent. I think that Saul Newman is certainly onto something in that there's a lot of exaggeration and fraud in these extreme longevity claims. That's something we've actually known about for a long time. I spent a dozen pages in my 1997 book talking about that. So that's a real problem, absolutely. However, the blue zones came from specific studies at specific times and places where they went to a great deal of effort to validate the longevity in those places. And one of the things that I think does not come out in Newman's work, which I think he believes if he can't find a government issued birth certificate, that there's no way you could validate an age. Well, there are other ways to do it. And for the blue zones, people literally wrote the book on how to validate great ages that were involved in that. Now, having said that, those things are evanescent in time. They may have been true at one time and not at another time. The good example of this, I think, is Sardinia, which is one of the blue zones. It wasn't Sardinia itself. It was 5 villages, distinct genetics. So I think that probably plays a role too. We think it's all lifestyle, but I think there's probably genetics there. Subsequently, those villages now have had more people move into them. Same with the others. The Okinawans, the young people don't live as long as the older people. My thought is maybe we could learn something from them, but I do like your idea of studying the longest live versus the shortest live people. I'll give you a good example of that. I live in Alabama, which is the 48th or 49th lowest life expectancy in the country. But we have a county here that's a real outlier. Shelby County is the wealthiest county here by far. It's one of the wealthiest 100 counties in the US. It also has the best educated people. It has the people that have the lowest rates of obesity and smoking and all this. If you put all these lifestyle factors together, that county should have a life expectancy on the order of what Marin County does. But it doesn't. It has a life expectancy that's just barely above the national average. So there's some local effect there that would be very interesting to tease out by studying, say, that county versus Marin County. What are they doing differently in those two places? I think I have a hunch It may have to do with the traditional Southern diet, but that's only a hunch. I think we could learn a lot about these things because the longest lived and shortest lived county in Alabama are 40 miles apart. What's the difference there? And again, the longest lived island in Hawaii, the longest lived county in Minnesota. I do think there's a lot to be learned from those, and particularly probably from specific even populations within those counties. Like one of the blue zones was this Loma Linda in California, but it wasn't really Loma Linda. It was really the Loma Linda Seventh-day Adventists because they had a specific lifestyle factor that made them a lot different than the other people that lived in Loma Linda.
I love Sardinia as an aside. It's one of my favorite places in the world. Alghero on the west coast in particular, and the province, the Nuoro County, which is the hotspot of centenarians, was on the east coast. And part of me wants to believe in the blue zones hypothesis. And also, if you go to Sardinia, many parts of Sardinia at least, it does feel like a very healthy place to be. People have a slow pace of life. Pedestrians always have right of way. It's very walkable. It's very beautiful. The cuisine is excellent. People don't get stressed out about things they shouldn't get stressed out about. And so I like the fact that you push back against that and what you said about how we need to bear in mind when the records were taken, and the fact that just because these places were blue zones back in the day, it doesn't mean they are anymore is really important.
Exactly. Yeah. I mean, they get westernized, they get fast food, they get lots of things. I have never been to Sardinia, but you make me want to go there, I have to say.
Oh, you should go. You should absolutely. And also, speaking about fast food, if I think about Alghero, there's no McDonald's, there's no KFC, there's none of that. People are proud of their tradition. It's just the loveliest place. But anyway, I don't want to get caught up on that. And I do actually want to return to comparative biology because you're one of the early people to push for studying companion dogs as a way to better understand aging, which I think has a lot of promise. And obviously, in the last few years, the dog aging project has gained a lot of attention, in part, I think, through things like podcasts, people like Matt Cablin speaking on podcasts about the dog aging project. Tell us a bit about companion dogs as model organisms.
Yeah, I've been advocating them for years. In fact, the dog aging project, I have to take a smidgen of credit for this,
Yeah.
came out of a symposium that I organized on companion dogs as aging models. And one of the people there was Daniel Promislow, who then went on with Matt to start that. But he wasn't interested in that in dogs before, but I showed him there are hundreds and hundreds of thousands of records on medical histories, longevity, and all of dogs, and they got very interested. So here's what dogs have going for them. I said before that our laboratory animals, one of their big drawbacks, besides the fact they're so short-lived, is that we keep them in such artificial conditions. Our companion dogs live with us, so they share our environment, and I think that's a really important aspect. If you look at the spectrum of diseases that they get, it's a lot more similar to the diseases that human gets. And in fact, there are breed specificities. So if you wanted to understand more about disease X, you could no doubt find a breed that was particularly susceptible to that. And then we have such good ways of assessing their health. We don't have very good ways of assessing mouse health. The mouse is either alive or it's very, very feeble or it's dead. But dogs, we have lots and lots of ways to test their memory, to test their physical function, because they can be trained to do sophisticated things. And then we have decades of veterinary records. So we understand a lot about the problems they get. So I just think they're wonderful. And then we care about them. That's the other thing, is that, if we can find something that can keep them healthy longer, very likely to work in us as well. But even if it didn't, it would be a huge boon for those of us who have animals that we love.
How would studying cats compare, out of curiosity?
Cats are interesting in their own right, but they have some unique features that I think make them less of a model than dogs. For one thing, they're carnivores. They really are carnivores. That is, whereas dogs, zoologically, they're in the carnivore family, but in fact, they're omnivores. They eat pretty much everything. And that has a lot of effects in their physiology. And the other thing about cats is they're not very trainable. For instance, giving them a cognitive test when they're older would be a lot more complicated than doing the same thing with a dog because they have their own mind about these things. Now, there are some things that they're very useful for. They have a kind of HIV. They have something very similar to HIV. It's called FIV. So they would be good for studying retroviral diseases. In fact, they're used for that. But dogs have cardiac problems, have problems with cancer. Some of them have lots of problems with cancer. Those are our top two killers. They have problems with arthritis. They have problems with obesity. They have lots and lots of things that make them very similar to us. So I think this is a huge resource that's just starting to be explored. It's not like dogs have never been studied in research before, but they tended to be laboratory dogs that were studied in the laboratory, and that sort of takes away, for me, some of their main advantages.
If we zoom out away from individual species, then in Methuselah Zoo, you comprehensively go through various different predictors of a high longevity quotient. What are some of those predictors? I know there are exceptions to these, but which are relatively consistent across different species?
A good predictor is the ability to fly. If you're a vertebrate, not so true in invertebrates, so insects are not so long lived. And I think that's part of making your own environment safer because you're off the ground and you can move, as you said before, you can move long distances quickly. Another thing is living underground. Animals that spend most of their lives underground or in caves, places that are safe. One of the things that I don't fully understand that I think is quite evident is that animals that live where it's cold tend to be long-lived. Now, for animals that are cold-blooded, being cold means that you're going to have a low metabolic rate, and that's certainly associated with long life. But there are animals like whales that are warm-blooded. The longest lived mammal that we know about is the bowhead whale. And that's also the whale that lives in the coldest part of the ocean. It doesn't migrate to warm water to breed like the other large whales. Mammals, when they get cold, that actually increases their metabolic rate. And then recently, we've discovered that bowhead whales and the other great whales probably have about 3 times the metabolic rate we previously thought they did. That's a unique combination of features. Something they share with the birds, which is a high metabolic rate but a long life. So those are the main things. Now, how living in the cold would be beneficial for whales, I have no idea. But in fact, all the longest lived cetaceans, narwhals are another one that are long lived, that live basically in the Arctic. I'm sure there are probably things that live in the Antarctic. Some of the Antarctic whales are probably also very long lived, but they haven't been studied to the same extent.
And thinking about all those different predictors, then we have things like hypoxia too. I know you speak about that in the context of naked mole rats, and then obviously those sheltered environments doing everything as slowly as possible, including reproducing as late as possible and moving as slowly as possible in the case of the Greenland shark. Right now, do you think that there's anything that we can practically take from these predictors in terms of our own human context?
Well, I think the hypoxia idea. So that's part of living underground, particularly for living underground in groups, because you're using the oxygen up and you're breathing out the carbon dioxide. So animals that live there tend to be very resistant. They tend to do well. Birds do well too in low oxygen. I think there's something to be learned from understanding how things adapt to low oxygen levels like that. we know quite a bit about that in humans from people that live in alpine areas, but I don't know that we know that much about it in subterranean living animals like naked mole rats, some of the other mole rats. But that strikes me as a pattern that's really worth investigating more.
It is interesting in the context of humans. I hadn't thought about this previously, but if you look at the different predictors of human longevity, then maximal oxygen uptake, VO2 max, is certainly one of the stronger ones. And that is one of the strongest determinants of endurance exercise performance. If you look at the levers that you can pull to boost your VO2 max, then altitude and the hypoxia that comes with that is certainly one of them. As an example of this, historically, a lot of coaches have tried to take a particular approach which involves living high and training low. So if people live up in the mountains, then that hypoxia can lead to some adaptations in their cardiorespiratory systems that can facilitate endurance exercise performance. And then training low means that the workloads they can accomplish in individual bouts is higher because they don't have that hypoxia. So the individual sessions can provide a stronger stimulus. So it would be interesting to look at how VO2 max varies according to the altitude at which people live, and then whether that is related to longevity. There are all sorts of confounding factors that you'd have to try to statistically control for, which would be very hard to do. I haven't looked at whether that's been done. Do you know of any evidence supporting that hypothesis?
A number of the places where people reportedly live longest do tend to be in remote mountainous areas. So assuming that some of those are really valid, it is quite interesting. And the relationship between VO2 max, certainly, and health in later life, I don't think there's any question that that's the case, whether it translates into longevity or whether it means that you're just going to die in a healthier state, which is something that's not a bad thing, is worth investigating. I just learned that a new way of training is using carbon monoxide, small doses of carbon monoxide, because that also makes you hypoxic. It's the equivalent of living at high altitude. So your body starts turning out more and more red blood cells if you give yourself periodic doses of carbon monoxide. They're trying to determine whether to make this illegal in the cycling world.
That's triggering the production of EPO, is it? And then in turn increasing.
Yep, exactly.
Interesting. I'm not familiar with that. If we look at animals with very different longevity quotients, do you think that there's an inverse association between an animal's longevity quotient and the scope to increase the lifespan of that animal? So these longer lived species, after controlling for their body size, if that is in part due to them existing in more favorable conditions, maybe because they can relocate, because they can fly, maybe we can't actually extend their lifespans as much as a result of that.
Yeah, I think that's very likely. And I think there's some evidence for that, which is that if you take small animals like mice that live 3 or 4 months and you bring them into the laboratory and treat them as well as we know how, you can make them live 2 or 3 years. So you can basically increase their life expectancy by 6 or 8-fold. We know that things like primates, when you bring them into captivity, do not have a very much longer life than they do in the wild. Some of them, in fact, are somewhat shorter lived in captivity, probably because we don't know the best way to take care of them. And then also, if you look even at the laboratory animals, we've learned how to make worms live up to 10 times longer. And if you look at fruit flies, it's 2 to 2.5 times longer. And if you look at mice, it's maybe 50% longer. And so worms are shorter lived than flies. Flies are shorter lived than mice. And we haven't so far been able to do much at all with non-human primates. You know, there was a 2 long-term studies of dietary restriction in non-human primates, and one of them there was a slight increase in survival, but the other one there was no increase at all.
Yeah, we might come back to that. There certainly seems to be quite a strong, and this is obviously just a hypothesis, but potentially quite a strong effect of the types of diets that those animals were given and how those varied according to the location. So the Wisconsin study versus the NIH study. In your book, Methuselah Zoo, you give some examples of animals that defy norms, those predictors of a high longevity quotient, living long lives despite shouldering certain burdens that you would expect to accelerate aging. What are some examples of these?
Well, I think the best example is the birds. And the birds have several things that if that's all you knew about them, you would think they would be short-lived rather than long-lived. Number one, they have a high metabolic rate. They have a metabolic rate that's about twice that of a mammal of the same size. The other thing is that they have levels of blood sugar that would be diabetic if they were in humans, but somehow they avoid the damage. And the third thing is they have a high body temperature. Their body temperatures are, 40, 41 degrees, so 106, 108 degrees Fahrenheit. That's a kind of fever that could kill a person if it were sustained. So if somebody told me, here, I have this animal, it's got a high body temperature, high metabolic rate, and blood glucose that's over the top. How do you think it's long or short lived? I go, oh, we're talking about something that's really short lived. But yet the birds live 3 times as long in the wild as same size mammals do in captivity. So I think this is an incredibly underappreciated and underexploited research project that, there are people now that are looking at naked mole rats and some of the other long-lived species, but there's virtually nobody who's working on the birds, which is ironic since the birds are so well known. A lot of them are so easy to maintain. I mean, people have maintained pet birds for centuries. I have 2 parrots myself, one of which is probably 75, 76 years old. My wife wouldn't let me experiment on that bird, I guarantee you. But it just shows that the resources are out there, the knowledge is out there for us to really dig deeply into why birds live so long.
Has anyone looked to see whether within the species, the birds that have lower metabolic rates and body temperatures and blood sugar live longer?
Yeah, and that's interesting. So no, there have been pretty much no study within species studies, and that's a really interesting topic because quite often the patterns you see between species are different than the patterns that you see within species. Within a species, large individuals tend to be shorter lived than small individuals. That's true in horses, it's true in dogs, it's true in mice, it's true in rats. I don't know if it's true in everything in humans. It's unclear. It looks like smaller individuals are less susceptible to some diseases, but there doesn't seem to be a clear pattern. But there's a lot of variation. Yeah, well, I mean, for people that have looked at different strains of mice and how they respond to dietary restriction, for instance, it turns out, or different individuals, it turns out that the ones that you can put on a diet that lose the least weight, that somehow adapt to keep on the most, those tend to be the ones that do the best, that live the longest. So the ones that can really adapt to new situations are the ones that benefit the most. Now, I don't know if they could do this actually probably in those primate studies that I mentioned, but I don't think they've ever done so.
Might be a retrospective analysis to be done there.
Right.
Focusing on the world champions of certain processes that are involved in the biology of aging. I know you've been studying those cohogs like Ming, trying to understand why they're so good at preventing the misfolding of proteins over time. I wonder if you could just briefly describe the relevance of this to aging, and then also just update how your research is coming on.
One of the things that we know that goes wrong with aging is that proteins, which drive all the chemical processes inside our cells, have to have a very specific folding pattern, kind of like origami, in order to perform their tasks well. But particularly for proteins that hang around a long time, they tend to be damaged over time. And when they're damaged, they tend to get sticky on the outside, and they tend to stick together and form clumps. And those clumps are at the basis of a lot of neurological diseases: Alzheimer's disease, Parkinson's disease, the plaques, the tangles. But they also are involved in muscle aging. And so when we were trying to figure out, well, what aspect of aging are these clams best to study? We thought the muscle. We decided that the best thing to focus on in the clams was this muscle that opened and closed the shell, that we would see how well, what was at the basis of that muscle's ability to avoid getting these protein clumps in it, which we know is detrimental to muscle. And so we basically squeezed the juice out of the muscles, and we used that, and we put a bunch of proteins in it to see if we could misfold them. There's various stresses you can do that make the non-white part of the egg, the white part of the egg that's clear, when you cook it and it becomes white, that's because these proteins have all aggregated. So we know a lot about that. So we would take all kinds of proteins and stick it in this clam juice and heat it, add chemicals that would supposed to make it misfold and aggregate, and none of them did. The clams were better than anything we tried. Only the long-lived ones, only the 500-year-old ones. We tried it with other species that live 100 years, 50 years, 30 years. And finally we thought, well, what about human proteins? So we took the protein that many people think is at the basis of Alzheimer's disease, the A beta protein, which is when it clumps, it makes the plaques. And we put it in the clam juice, and we did the same thing in the equivalent juice from human muscle. And it was very protected against these things aggregating. So the next thing we wanted to do is try to figure out, well, how does it do that? Is there some secret molecule in there that's behind this? And so far, we've discovered many things that it's not. So we tested all the known things that prevent or resist protein misfolding, and we demonstrated conclusively that it's none of those. So there's some mystery molecule or molecules in the clam muscle that somehow is resisting this. We don't know what it is. One of my former graduate students, who's now at Harvard and a postdoc, he's the one that started that project. And when he left the lab, I said, this is a good project for you to take with you. So he's been working on it. Hopefully he will solve it, come up with some magical therapy for Alzheimer's disease, get fabulously famous and rich, and I'll be very satisfied with that.
This is a non sequitur, but one of your papers that I particularly enjoyed reading was about the ubiquity of antagonistic pleiotropy. And when I say about the ubiquity, it was asking whether that is indeed the case. And I wondered if you could just touch on what antagonistic pleiotropy is, because we've glossed over this a couple of times already, but I think it's a very important concept to understand when discussing the biology of aging. So,
Sure.
what is and how ubiquitous do you think it is?
Yeah, so pleiotropy is just a word for multiple effects. Genetic pleiotropy is that certain genes have many effects. Antagonistic pleiotropy is the concept that genes will have beneficial effects at some ages, but will have detrimental effects at other ages. And this is thought to be a key factor sort of limiting longevity, is that genes that are beneficial early on, when animals are most reproductive and leaving the most offspring, may be detrimental later on, but no matter how detrimental they are later in life, nature will still favor them if they're so beneficial early in life. This is an old idea. This idea stems from the 1950s, but nobody had any genetics to try to really test it in the 1950s. And George Williams, who really developed the idea, imagined this process by which calcification in bones, rapid calcification bones, is beneficial early in life because it leads to strong bones early on. But the continuation of that process later in life could lead to calcification in arteries, which is clearly bad. But again, this was just a hypothesis, and he had no notion. Well, now that we have so many ways of making animals live longer, either with diets or drugs or genetic tinkering, we can look to see, well, what's the evidence? So genetically, we know about at least a dozen genes that if you knock them out or partially cripple them, make mice live longer. In every one of those, there are early life detrimental effects, because the opposite of this idea that there are things that are beneficial early in life but detrimental late in life is that if you can make things less detrimental late in life, you're likely to make them less beneficial early in life. So, for instance, there are a number of mice that live longer, much longer, if you partially disable or completely disable the activity of growth hormone. If you do that, though, it delays their maturation and either makes them sterile or makes them very feeble at reproduction. And we looked at a number of cases in worms and flies and mice, and every time we found a gene that seemed to make them live longer, it seemed to have some early life detrimental effect. Detrimental in terms of reproduction, not necessarily survival, because let's face it, nature is all about reproduction. The one that leaves the most offspring, those are the genes that will come to dominate in the population.
It's interesting to think about it in the context of our obesogenic environment, because the total amount of energy that your body has available is going to influence the onset of your reproductive life. Kids that put on more fat tissue are likely to go through puberty earlier, enter reproduction earlier, and they could therefore be more reproductively successful. There are other factors that are at play too, like sexual selection and so on. But it does just make me think, and there are many different directions in which we could take that conversation. But I wonder if you've got any brief reflections on that.
Yeah, I mean, it's an interesting idea. We know that as we've gotten better at preventing diseases and have gotten better at producing nutritious food, that the age of puberty has been declining. It's been declining for a century and a half.
Yeah.
And yes, in a population that didn't have contraceptions, that would have certain advantages. Probably doesn't today, because people you typically reproduce now for the most part when they want to, at least in the developed world. This is all different, of course, in places where contraception isn't so available. But I think a lot of our increased longevity has to do with the fact that we've gotten so much better at nutrition and avoiding a lot of serious childhood diseases.
I never thought about that, but maybe the use of contraception is one of the biggest boons to human longevity. The
It
experiments
could be.
Michael Rose, I know there have been these experiments where scientists have delayed the point at which fruit flies can reproduce, and then when you do that, they do actually live longer. So if you're using contraception, and you're having babies later as a product of that, you're pushing back the age at which you're going to conceive, and then over generations that might be expected to extend human lifespan. So maybe the condom, the ultimate.
Yeah, I mean, we might be selecting for later and later age at menopause now, but it's slow. It would be slow in humans, because one of the differences between menopause and Michael Rose's experiments is that he was preventing them from reproducing at ages where they normally were very productive. This is one of the interesting features of menopause. It's not like shutting off a tap. It's that fertility tends to drop from about the age of 30, and that's just kind of the end of it. But now that birth patterns are changing, women are having babies later and want to have them sometimes even later than they can currently. And now we have all kinds of technology that helps do that. It's going to be interesting to see what happens to the age at menopause, just naturally.
One implication of antagonistic pleiotropy is that if one of the side effects of gerotherapeutic interventions that are targeting some of these genes or their protein products that are antagonistically pleiotropic, that might well affect reproduction early in life. So do you think it will be best to take these gerotherapeutics in the post-reproductive years if people still want to have kids, emphasizing the importance of the age at which you start taking these?
Yeah, I think that's very likely to be the case, and I think that's likely what would happen anyway. People in their 20s typically are not going to worry about aging so much as people in their 40s and 50s. And from what we know, one of the surprises of the drugs that we now know can intervene in mice, at least, is they seem to have beneficial effects even if they're started relatively late. And I think that's a surprise because for decades, the only thing we knew to make animals live longer was dietary restriction. And with dietary restriction, the longer you did it, the bigger the effect. So it was most effective if it was started right after weaning. But with these drugs, you seem to get just about the same effect if you started at the mouse equivalent of 60 years as if you started at the mouse equivalent of 40 years. So that's one of the really positive things to come out of this, is that even if drugs like that had antagonistic pleiotropic effects on reproduction, because it can have effects on other things like muscle growth, if you simply avoid doing it until you're done reproducing, then you avoid that problem.
Saw a paper yesterday, recently published, suggesting that intermittent fasting, and I forget the schedule they used, it might have been a 24-hour fast every 3 days or so in mice, did confer some health benefits in adult mice. But actually, when it was implemented early in life, it led to some beta cell dysfunction.
Yeah, I mean, there are these interventions that are very brief, but happen at a key period of development that do seem to have long-lasting effects. So, for instance, the life extending effect of growth hormone on mice, of reducing growth hormone, can be completely obliterated if you just give a little bit of growth hormone in the first 6 weeks of life. Then it doesn't matter if they don't have any growth hormone after that, they still are short-lived. So there may be these windows very early on. I would be very concerned about that approach just because so many other things are going on at that point in life. Growth of the brain, growth of the muscles, growth of all kinds of stuff, that I'd be afraid that there are things that have unknown effects. And the other thing is that in the human case, people of that age, it shouldn't be having decisions made for them that might affect them much later in life. For instance, I just can't see, oh, little Johnny, we're going to fast you 2 days a week because we think that, when you get to be 60, you'll be much healthier than you are now.
Little Johnny would find a way to get to the convenience store. And
I think you're exactly right. That's right. Yeah.
In the last decade or so, a lot of the geroscientific community has focused quite heavily on the so-called hallmarks of aging. And you yourself have published work on several of these: proteostasis, and with respect to the hallmarks of aging, that's loss of proteostasis, mitochondrial dysfunction, and more. And my impression is that focus on the hallmarks of aging has somewhat hamstrung progress that could otherwise have been made because everything has been couched in those terms. And meanwhile, if you look at how those were developed, the initial paper was just a narrative review paper. It wasn't based on some sort of systematic analysis of the determinants of aging by any means. I wonder what you make of those, and also how you might go about better identifying them.
Well, I mean, I think the hallmarks are a good list of
Yeah.
things that we know that go wrong with aging. The original hallmarks, if you looked at the authors list, you could almost see where they came from. Oh, yeah, this author works on this, so that's one of the hallmarks. This author works on this. But they were a reasonable list. And then they got expanded after that. They added 3 hallmarks. I don't think they're anything more than a list. But what surprised me is they so quickly went from being a list to being some sort of scripture. This is the way to study aging, is to investigate these hallmarks. Well, the interesting thing, I think, is the interactions among the hallmarks. And then there are going to be processes. I mean, the list grew from 9 to 12. There's going to be new things discovered. The thing is that they're all interconnected. And I think by just saying, well, you have to focus, as some granting agencies do, they expect you to focus on specific hallmarks. That's not how the hallmarks were discovered. The hallmarks were discovered by individual investigators pursuing what they thought were interesting ideas. And I do think that an over focus on those and a sort of avoidance of research that might look at something completely different is a mistake. And so I do think that they haven't been all that useful. You can cite those papers in your grant proposal, but again, it's a list. And yeah, I'm glad somebody put that list together. It's a nice, concise place to find out. But as I said, it's the interactions among them rather than the individual hallmarks that I think are the keys to understanding aging. And just by focusing on this one or that one, you might be missing the big picture. Well, how does this one affect that one? And how are both affected by this other process that we didn't realize was a hallmark, but we just discovered, like the microbiome. That was one of the ones that was not in the original group. Now it's in there. Now we know it does a lot of things we didn't appreciate before. If the hallmarks had come out 10 years earlier, we might not have discovered what we know now about the microbiome.
And them having these network-like properties means that if you affect one node in the network, then you tend to affect perhaps all the other nodes in the network.
Exactly. Biology is complicated. As I tell people, I said, if biology was simple, it would be physics.
Are there any ones that you think should obviously be added to the list that are clearly missing that have good experimental support?
That's an interesting, I haven't, since I don't tend to think in terms of hallmarks, I don't tend to think in terms of, well, should this be added or should that be added? I know that before the latest version of it came out, there were a lot of people saying that, oh yeah, the microbiome should be added. One thing that's, I think, very much underemphasized is the interaction between cells and between tissues. There's one of the hallmarks that basically is intercellular communication. Well, that's very vague, and it probably incorporates a ton of things, from hormones to exomes to these things that cells are spitting out, all kinds of stuff that. So to a certain extent, by having these broad categories like this, you're sort of lumping together what might be a lot of really interesting subcomponents of it.
Yeah.
I mean, the thing that has come out that we didn't know before is there are these little short peptides, that there are thousands and thousands of them being produced by the genome now that we had no clue about before. Could be that micropeptides is going to be the next hallmark. It could be that exomes is going to be the next hallmark. This is why I think you don't want to limit yourself to thinking in terms of it never enters my mind unless maybe when I'm writing a grant proposal to think about, the hallmarks. I just think about, well, this is an interesting process, but yeah, so there's just 2 that I came up with that are new. I hadn't even thought about it before. But yes, what if these micropeptides turn out to be driving all kinds of things that we don't appreciate yet?
Altered intercellular communication is an interesting example because, like you say, within that you can put paracrine signaling factors, you can look at endocrine signaling. Are we speaking about hormones that act over long periods, or cytokines that have briefer
That's
effects, or
exactly.
neuron
And this is
signaling?
one hallmark, right? This is one hallmark that all of that is subsumed under.
Something that I've suggested on this podcast already is that if we are thinking about this in terms of hallmarks, then I think there's very strong support for the inclusion of clock dysfunction in there. When I say clock dysfunction, I'm not saying circadian dysfunction specifically, because we have biological rhythms that occur across different timescales. But if you look at the criteria for what constitutes a hallmark, then those include conservation across different species. And certainly there are exceptions to that when you look at some of them, like in your book, you point out the fact that naked mole rats can bear a massive burden of oxidative stress in their tissues and live very long lives despite that. But there's that. And then obviously, if you amplify the hallmark in question, then you accelerate the organism's aging and curtail lifespan. And if you slow it, then you decelerate aging. And there's good evidence in support of dysfunction, whether that's through extrinsic factors like aberrant light dark cycles or mistimed feeding, or whether it's through intrinsic factors like genetic disruption of the core clock machinery. What we don't have so much evidence in support of is whether improving the function of the clock does extend lifespan. But as we might come to later, there is a bit of evidence in that direction when it comes to appropriate use of feeding timing and how that interacts with, say, caloric restriction. But I want to move on from the hallmarks to biomarkers of biological aging and biological age. You've done some work on this in recent times. I really enjoyed a paper that you published in one of the PLOS journals that was looking at biomarkers of resilience. And obviously, if we can develop biomarkers, then we can accelerate studies on candidate gerotherapeutics. And you've been quite vocal about how studying resilience could be very helpful when it comes to developing these biomarkers. So could you touch on that paper that you published last year, what you did and what you found?
So there we were trying to use resilience itself as a biomarker, or animals that are more resilient. And by resilience, what we mean is the ability to resist and recover a stress. And the stresses that we wanted to use were stresses that we thought would be translatable to people. Let's say if something recovers from surgery faster, or is that going to tell us something about their longevity? And what we found surprisingly, so the stresses we gave a surgical stress, that is anesthesia. We did wound healing. We did response to a blood draw. So how fast can your body replace the red blood cells if you removed one? And we did response to a pathogen. And then we did that in giving animals therapies that were long since demonstrated to make them live longer, either dietary restriction or 17 alpha estradiol. And what we found to our surprise, this is why you do the research, not just think of the ideas, is that we found really the opposite, that the ones that had received the gerotherapeutics seemed to respond less well, if anything, in our resilience assays. So, in a certain extent, that kind of result is disappointing. To another extent, we learned something from that. And I think one of the things we learned, one of the lessons I took away from it, is that the environment in which we keep our animals is having a big impact on how we think about therapeutics and how we think about aging. And we really shouldn't expect that animals that their whole lives have had the best food, the same temperature, the same light regime, all this stuff, are going to behave in the same way that we might expect animals living in a more realistic environment might do. Animals that have been protected from any pathogens. Now, when we did that experiment, I hadn't yet come across this literature about how the specific pathogen-free environment cripples the immune system. So I didn't think about that. But now that I'm looking at our results, And I'm thinking about that. I'm going, well, yeah, okay. These animals, they had a crippled immune system. What could we gather from that? The dietary restriction and the 17-alpha estradiol, they're not going to fix their crippled immune system, which requires exposure, to other microbes. So to me, the valuable lesson out of that is that we really have to rethink the way that we test our drugs. I don't think we do it the right way. And that's one of the reasons why 95% of successes in mice don't translate to people, is that we're just not going about it in the right way.
One complicating factor that I think of when considering this approach is that there are going to be large variations in responses depending on what you look at. So if you take the example of how people respond to exercise training, then you have people who might respond very strongly when it comes to one thing, so say VO2 max, but when it comes to something else, say insulin sensitivity, they might not respond as favorably. And obviously, exercise is a stimulus that, given supportive conditions, confers all sorts of beneficial effects. But if we think about testing resilience through some other things that you didn't use in that specific study, but you have mentioned previously, like how people respond to sleep deprivation, then based on the relatively little research that's been done so far on this subject, if you look at, say, cognition, there are some people who might have their working memory strongly compromised by sleep deprivation, whereas measures of attention aren't so negatively affected. Or it might be that one person maintains their cognition relatively well, but their metabolic regulation goes out the window and they become much more dysglycemic than others, their peers in the same group. So I suppose it really depends where you're looking.
Right. And I think this points up one of the big limitations of our research is if you look at humans, we have all this genetic variation. And on top of that, we have all this environmental variation. So we are doing some studies now that, for instance, we have some rats we're looking at. These are genetically mixed kind of rats, but they have one of two different mitochondrial genomes that differ in quite a bit in the genomes. And what we found is that one of those mitochondrial genomes seems to be associated with better endurance performance. And the other one seems to be associated with better strength performance. And, I say that, but it's not surprising. And just look at the mitochondrial variation we have among humans, right? And among mice, there's almost none. The standard laboratory mice all came from a small group of ancestors. So if you look at their mitochondrial genomes, they differ, that's 16,500 nucleotides. They differ in 3 to 5. Humans differ in dozens and dozens of these things. So this is not a surprise. This is one of the limitations that I think in our testing of drugs. We need to use a greater variety of genetic backgrounds, a greater variety of environments. I mean, we don't want something that doesn't work if you get warm. We want something that benefits organisms under pretty much all conditions, because humans are experiencing pretty much all conditions.
And if we think about humans, are there any more accessible quotidian proxies of resilience that you think might be helpful for us to look at? Just as an example of this, you could look at how quickly somebody's heart rate recovers towards baseline after a bout of exercise. Is there something that we could look at on a regular basis that isn't so stressful as sleep deprivation or some sort of immune challenge to get a proxy of how resilient we are?
Things like that could certainly be looked at, but they would only apply to people in basically probably that ever do any exercise. As somebody that's been sitting on the couch for the last 30 years, my guess is you would learn relatively little from them relative to people who were regularly physically active. Maybe you'd learn more actually from the people that never exercised. Maybe the ones that never exercised that had the quickest return to, if you could get them to exercise and not die, that had the quickest return to their base. Maybe it tells you more than it would for people who are already somewhat trained. This is the thing about people, right? Is that it's not just the genetics; it's the lifestyle, and maybe the lifestyle that you did before you were 10 years old that you have relatively little control over.
Conscious of the clock now, and you've been very generous with your time, so thank you for that. And needless to say, we've got through about half of the subjects that I wanted to touch today, but I could be quite long-winded and tangential, so it's no great surprise. I thought we'd end with some quick thoughts on a couple of subjects. One of the subjects, expanding on what we were just discussing, is some of the other ways that we might look at somebody's biological age and biological aging. Obviously, a hot topic nowadays, and we've seen the commercialization of various different products trying to assess biological age and pace of aging. What do you make of some of these omics technologies? And are there any that you think are particularly promising?
Yeah, so all the omics that are trying to assess biological age, I think they're very interesting. I think it's also quite preliminary. My guess is that there may be no such thing as biological age, to the extent that I think our different organs are likely aging at different rates, depending on our genetics, depending on our lifestyle factors. And it may be some way that you can say, okay, well, let's synthesize all of that and come up with one number. But it seems to me, from a therapeutic standpoint, what I'd really like to know is, how is my heart doing relative to other people's heart at the same age? And how's my brain doing? How are my kidneys doing? And I think that's all likely going to be discoverable from proteomic and metabolomic investigations once we know enough about it. The translatability of that, I'm just not sure, because that's very costly at this point to do. And I recently heard a talk by someone who's planning to do just a massive analysis of thousands and thousands of people, but it was going to be millions and millions and millions of dollars. And I asked him, well, is this ever going to be something that's available to everybody? And he said, well, yeah, it's like DNA testing, right? It was $3 billion for the first human genome, and now it costs less than dinner to do a human genome. And I said, well, okay, well, maybe if that's the case, then that'd be great. But maybe not, because it not only requires a great deal of biochemistry, but it also requires a great deal of computation. At some point, I think the cost can only go down so much. It does raise an interesting point, though, which I think that gerotherapeutics, as they develop, are going to have to be tailored to the individual. I don't think giving everybody rapamycin or everybody metformin is going to be the way to go. We're going to have to figure out, and then what are your side effects, and what do we need to do to counter those side effects? Because, even the GLP-1s, there's a lot of people can't tolerate those. Clinicians tell me a third to a half of their patients can't tolerate those. Maybe we'll figure out better ways, or you take these other medications to help you tolerate this. Ultimately, it's going to be personalized, and, with everybody's genome so accessible now, and probably their epigenome is going to be accessible pretty soon. I think that what we need to look at is this is right for you, and this is not right for you. We already know that, right? I know people try ketogenic diets who think it's the greatest thing in the world and lost 100 pounds, and other people, they tried it a week and they said, oh God, I have no energy. I hate this. We already know that this kind of variation is out there. We just have to learn how to deal with it.
Something that you've mentioned today, but spoken a lot about previously, is that it might be that combinations of these different gerotherapeutics are best rather than single substances, not just to manage those side effects, like you said, for some other reasons too, but there's been very little research on combinations of these. Do you have any brief thoughts about how you might try to model the interactions between these different substances? Because there are so many supplement companies out there, for example, that are combining different natural products. Maybe they're using some sort of combination of nicotinamide riboside or nicotinamide mononucleotide with some potentially senolytic substances, with various other things too, hoping that these things have additive effects when some of them might actually antagonize each other. How might you try and model the interactions between those different things?
We need to do the research, and we need to start off doing the research in experimental animals. But one of the reasons it hasn't been studied more is it's hard to get it funded. And the supplement industry has no motivation to find out if it really works or if they're really antagonistic, as long as they can sell their products. And I worry about that, because you're absolutely right. It might be that senolytics put together with NAD enhancers are antagonistic or are even somewhat pathogenic in combination. It's not obvious why that would be, but that's why you do the research, because things, aren't always going to work out the way you think they are. That's the way science moves, is when they find out, oh, this is what we all thought, but we were wrong. It's actually this way. So I think there's a huge need for investigation of these kinds of combinations, and maybe they're synergistic. That would be great to find out, but we'd like to find out before people start taking massive amounts of them. It's like the Rapamycyin self-study group. They're taking different doses and different regimes, and we're not going to learn anything from them because it's totally uncontrolled. And we'd like to know more. I personally would love to know more about the effect of rapamycin in people. That's going to take controlled studies, and somebody needs to find it. And the thing that worries me is it's very hard to fund studies that somebody's not going to make a fortune on, depending on how the study comes out. That's why the metformin TAME study has never been funded. If metformin turns out to be this miracle drug, nobody's going to make any money. It's as cheap as aspirin. That's the climate that we're in right now, and it bothers me a bit, I have to say.
And of all the different things that have been studied for their effects on lifespan and proxies of health span, calorie restriction remains the best studied. We could easily do a several-part series of podcasts about calorie restriction by itself. But if I had to pick one question to ask about that subject, then it might be, when it comes to calorie restriction, do you think that if it is having positive effects, then those are largely mediated by positive effects on body composition? And then obviously any effects of calorie restriction are going to interact with someone's baseline body composition. So we spoke briefly about the Wisconsin and NIH studies on non-human primates and how those responded to calorie restriction. And in one of those studies, the diet was probably substantially more obesogenic than in the other study. And sure enough, it was the study with the obesogenic diet where the restriction had some positive effects. So if we think about the human context, then calorie restriction might confer some health and longevity benefits, but probably only if somebody is carrying an excess of adipose tissue at baseline. Relevant to this conversation is also the amount of fat-free mass that someone has. If somebody has sarcopenic obesity and they go on calorie restriction, they lose some fat mass, but they lose some fat-free mass too when they already have little muscle tissue and low bone mineral density, then maybe for that person, it's not necessarily where you start, and you instead start with resistance training and focusing on not losing weight necessarily, but building more fat-free mass, and along the way you're going to lose some fat mass as well. So just to reiterate the question, is it changes in body composition that are doing much of the heavy lifting?
I think that's part of the story. The changes in body composition, I think, are part of the story, but I think they're only part of the story. And the reason I say that are the many health benefits that seem to accrue from time-restricted eating, which has many of the same benefit as calorie restriction, but doesn't necessarily have the same effects on body composition. And the other thing is people can do it. We've now done enough dietary restriction stuff in people to realize that people can't really stick with that. But time-restricted feeding, they can. And so I think the other part of the calorie restriction effect is this fasting physiology that comes from periods of 12, 15, 16 hours of fasting, which does change all kinds of physiological processes in the body. And so I think that some of these new dietary regimes, the alternate day feeding or time-restricted feeding, or some of those, I think some of those are going to be highly beneficial. And like I say, the thing about it is that people can do that. The other thing about calorie restriction, you're right on, is the muscle loss. For the people that really do it, and the people that I think really do it are the people that were in the calorie restriction society. They lose a lot of muscle and they lose a lot of bone density. And those things are not good when you get older. One of the problems with the GLP-1 drugs for people taking them, because you put the weight back on when you stop the drug, well, particularly for older people, they lose the fat, but they also lose the lean mass. And then they come off of when they put the fat mass back on. But it's hard to put on lean mass the older you are. And so you could see, well, they put the weight on, then they go back on it again, and they yo-yo on and off. And every time they're losing more muscle. And that is just a bad scenario. But I do think all these new diets do have some real promise, and people can do them.
I completely agree with everything that you've just said. Despite my question, I don't think that it is body composition that is mediating all of the beneficial effects. As I've discussed on the podcast previously in a couple of episodes that I did on chronobiology, I do think that time-restricted eating is very helpful. Initial clinical research shows that, and I'd argue that probably the best of that has been done by Courtney Peterson and her colleagues. So people can refer to those episodes if they want to hear more. I also think that, as I touched on earlier on, much of the calorie restriction research on non-human animals is probably confounded by time restriction. And by far the best demonstration of that I've seen is work by Joe Takahashi and Victoria Costa Rodriguez, where they use these automated feeder systems to show that mice will self-impose time restriction when they're given access to a low-calorie diet round the clock. They'll consume it all within 2 hours or so. And then they published a subsequent study about 5 years later showing that if you calorie restricted the mice and you did so by giving them food pellets at scheduled intervals around the clock, then that did extend lifespan, extended median lifespan by about 10%. However, if that was combined with time restriction to the active phase, so the periods when the animals would naturally be awake and eating, then there was an additive effect on lifespan extensions such that the animals lived about 35% longer. And that was without any change in body composition, and some of that related to changes in patterns of gene expression. So that's suggesting that the circadian system might mediate some of these beneficial effects. And then when it comes to losing fat-free mass, something that I'll mention here that I think is missed by a lot of people is that while a lot of these studies, and this includes the calorie study, are focused on measures of relative strength, so how strong you are relative to your body weight, absolute strength matters for activities of daily living too. If you just think about opening a jam jar, for example, or lifting something to put it on top of a wardrobe, whatever it might be, the absolute strength that you have is important too. And so one of the issues that people might face if they're losing some fat and they're gaining in relative strength or maintaining their relative strength but losing absolute strength is that their ability to perform these activities might at some point become compromised. Just to wrap things up, Steve, I'll ask one final question about aging, and that's a big picture question. If you think about current trends in the world, and those include things like changes in the climate, how do you think the determinants of aging might change in years to come? And I realize I've just primed you with mentioning the climate, but obviously there are lots of powerful forces that are at play. The world is constantly changing. And so just because something might have proven beneficial in 1980, it doesn't mean that it will in 2040.
Yeah, well, interesting question. I would say that the trends in the developed world are very different from the trends in the developing world. And in the developed world, the trends are antithetical, I think, to longer health. Thinking of climate change, I'm thinking of dietary changes, I'm thinking of more and more obesity. In the developing world, I think the opposite is true because they're getting better nutrition, better medication. Ultimately, they will face the obesity issue. And so, there's some people that worry that we're going to develop gerotherapeutics that are only available to rich people. And so we will develop a sort of two-tiered, society, the people that are living a long, healthy life and the people living a nasty, brutish, and short lives. I think that's quite likely, but I also think that that's pretty much where we are now. If you think about it, the people that I know, that they go to doctors regularly, they exercise regularly, they're going to live a lot longer than the people that are struggling to make ends meet, that have to get up and work several jobs. That's unfortunate. I think we ought to really think about how we want our society to evolve. If I wanted to, for instance, if I wanted to increase life expectancy in my home state here of Alabama, I would completely ignore anybody with a college degree. They already are going to live a long time. I would look at the people that are suffering from obesity, diabetes, all these things at an early age and say, okay, well, we need to fix them. That's going to have a much bigger bang for the buck than making university professors live a little longer, stay healthy a little bit longer.
I do think that we really need to focus on the democratization of longevity. It's fine me saying that as a relatively privileged white person, but I appreciate your answer for that reason. And I do worry that a lot of these gerotherapeutics are just going to be snapped up by rich people. And obviously, in recent years, there's been this increasing privatization of geroscience. And that concerns me in many ways, especially with the political situation in the US and then how that might shape funding for research by academic institutions in the US, which have done more than anywhere else to develop research in this particular sector. And also, I think it's interesting to consider the fact that if more and more people are being snapped up by these types of businesses, and that's diverting talent away from universities where people probably would otherwise get the best training, how is that going to dilute the quality of research in years to come, as well as any potential progress we will make? But that's a subject for another time. And needless to say, I'd love to discuss that with you at some point, but might have to wait until you've written the next great American novel to have you back on. Thank you so much, Steve. And I'll just end by asking, is there anywhere in particular that you'd like to direct listeners to learn more about your work? I will at this point plug your book as hard as possible because I love your book, Methuselah Zoo. But I know it's one of many books that you've written. And I also appreciate the fact that you try to communicate this progress to the general public through many different avenues. You're not just a siloed university professor. And I think a lot of that is a product of your unusual background.
It could be. And I thank you. I don't think there's a single place, but I think there are some good podcasts that people should pay attention to. And yours is certainly one of them. People need to be discriminating, I would say, because there's a lot of more and more erroneous, giddily hopeful ideas out there that have no basis in anything that we now know scientifically. So, keep up the good work is what I would say to you, Gregory, and it's been nice talking with you.
Yeah, it's been an absolute pleasure. Thank you, Steve.